Thank you very much to Helen for reading my title for me. It's quite a long title. I'm a clinical lecturer in Manchester, and this is our planned study from Badbeer. And I guess it's nice to have the next slide there. This is a slide that many of you might be familiar with, an example of these treatments called biological treatments. They're mainly injection treatments. They're complicated treatments. They're treatments made out of antibodies, which are big proteins that mimic the same proteins that we have in our immune system. And on the left here, you can see the original adalimumab, which is called Humira. That's kind of the daddy brand of adalimumab. And then you've got all the son and daughter brands now that came onto the market in 2018 on the right, and we generally call these biosimilars. So you can see they're all called adalimumab, but they have different brand names: Imraldi, Amjevita, and Hyrimoz as example. Now, I think to understand the whole situation, we have to go back to what biologic therapies are. And so the next slide is a nice kind of summary of what these molecules are. And I think it really illustrates the fact that these biologic therapies in psoriasis, for the treatment of psoriasis, are huge molecules. So you can see that aspirin is tiny, 180 Daltons, that's measuring kind of the mass of these molecules. Insulin and erythropoietin are both different biologics. They are still biological treatments, so they're made from cell lines similar to the ones that we use in psoriasis. But even compared to a monoclonal antibody, which is the ones like adalimumab and the ustekinumab that we're familiar with for the treatment of psoriasis, they are a lot smaller. So you may ask, so what is the significance of this, the size? And I think the significance of this is it makes the treatment more complicated. And so it is a lot easier to reproduce aspirin in a lab. You can know the chemical structure and then reproduce it pretty reliably. But with biological treatments, it's a bit more difficult. It's grown from cell lines. You have to work to find the right cell that could produce this particular antibody. You need to make sure that the active areas of this big molecule are similar or the same. And really, a biosimilar is defined as these have to be clinically the same. They have to act in the same way. They have to have similar properties, reliable properties that are similar to the original product when it's used in human beings and in use in labs. And therefore, it is not as easy as producing, for example, paracetamol for the supermarket. And the next, this figure really kind of demonstrates some of these differences. So the actual protein structure of the antibody needs to be the same. So we call these amino acid chains. So these kind of combine to make a protein. And the circles represent, for example, the different sequence that you would get in, let's say this is adalimumab. So both of these are adalimumabs. The chain coding for adalimumab is completely the same. But because they're grown from different cell lines, different structures, they have slight subtle differences. So there's something called glycosylation, which are stuck on bits of sugar basically on the protein. And these stuck on bits of sugar might be a bit different. And these may affect on how this protein is handled by the body, how this protein unfolds, distributed in the body. And therefore, these might have an effect on how the treatment affects the individual and treats the psoriasis. And a nice little analogy is one of cakes and sugar. So aspirin is a little bit like the sugar. So sugar is quite a simple thing. Sugar from one brand and another brand, they're pretty similar. You probably won't tell the difference if it's the same granulated sugar. So they're pretty similar. But a biologic is a bit like baking a carrot cake. There are different recipes, like different cell lines. So both of these are carrot cakes. One is maybe from Waitrose, one is from Tesco. The main ingredients of a carrot cake is the same, so they both have to have carrot to be a carrot cake, but they may have different levels of sugar, different levels of icing. This one seems to have a bit of pistachio on the left, the right one has a bit of walnut. So this is analogous to the biologic kind of and the biosimilar situation. And so what is the problem, or why do we need to look at this if they're pretty much the same? And I think one example illustrates this nicely. So as I alluded to earlier, biologic therapies are not restricted to inflammatory disease or psoriasis. They've been used for many years for smaller molecules. So this is EPO. This is a biosimilar of EPO. And epoetin is a treatment doctors would use for patients who have anemia or low red blood cell count. For example, after chemotherapy and so on. And it's really important because these patients can have a very low count, which makes them breathless, and giving them EPO stimulates the bone marrow to produce red blood cells. But in this particular biosimilar, what happened is that this one was linked to a condition called a red cell aplasia. And that essentially meant that instead of treating anemia, this product actually gave the patient anemia. So you can imagine that this is quite a big deal. Because of these subtle differences, this can cause this immune reaction, causing a completely opposite effect. Now, we have no evidence at this stage that the biosimilars we use in psoriasis or inflammatory disease have any problem as big as this. But when I had a Google, this is a nice little blog by a patient who has Crohn's. And here, the title is, "I'm all for saving NHS costs, but this biosimilar medication just isn't similar enough." And you may know that the big advantage of biosimilars coming onto the market is that it's really helped the competition. It's really lowered the unit costs to the NHS. And potentially that is really good news. Because that means that more people can be treated with the same pot of money, and in public finances, that's great news. More people can get hold of the treatment. But in this blog, this patient is talking about her experiences, her NR1 anecdotal experience. And what happened with her is that when she was switched from Humira to Amjevita, which is one type of biosimilar, and as you know, on the NHS, these decisions are made on a regional level, a commissioning level, because of price. She experienced some side effects, which made her wonder, is biosimilars actually similar enough? And what you might not know is that biosimilars can get approved without the clinical studies in all of the indications. So for example, let's say one of the adalimumabs, they run a study in patients with Crohn's disease, and they demonstrate that, let's say it's Amjevita, Amjevita has the same effect for Crohn's disease compared to Humira. And if they measure in the blood, it's very similar. So it's the mechanism of how it works in the body, it looks very similar. Then the FDA and the MHRA then say, yes, this looks very similar. We're happy to go, let's approve this. So this means that some of the biosimilars we use, for example, in psoriasis, may not have been trialed particularly in psoriasis, patients with psoriasis. And so this may be a problem. This may not be a problem. We don't really know. So this is why it's really important to monitor and observe what is happening in the clinic, what is happening in patients who are getting these treatments. And so this comes to the main question of this PhD studentship. And the question is a little bit like being Dr. Strange and asking ourselves two time-related questions, really. So the first question is, what would have happened to patients had the switch to biosimilars not occurred in people who switched? And that's a little bit like a Dr. Strange question of going in time and then going back in the past and then going forwards in time again with another scenario. The second question is, what would have happened to the patient if they started on a reference product instead of a biosimilar? So that's relevant for people who started a different product, so Amjevita, let's say, compared to Humira. And again, this is a kind of a time question. We want to try and ask these what-if questions, and these questions in science are called counterfactuals. And so to do this, we need to have first of all, different groups of patients starting or switching these treatments. And Helen has kindly read out the BADBIR acronym, so I don't have to go into this, but many of you may already be familiar with BADBIR. This is a multi-center observational study, a registry, really monitoring drug safety in patients with psoriasis. And we have 3 different cohorts. People get registered to one of them, and if they switch to treatment, they go on to another cohort. So we have the conventional cohort, patients who are on the traditional treatments like methotrexate. We now have a newer small molecule cohort of patients on Premalas, and then the biologic cohort, which is very full. And you can see here, we've highlighted a couple of the treatments that we're particularly interested in this study. These are the biosimilar brands, and we're going to compare them to the originator brands, which is Humira and Enbrel, which is we still have data for. So how are we going to answer those Dr. Strange questions? Well, I think first of all, it would be really important to know what is happening in the UK. So the first study really is a study to map it out, to look and see what the usage and uptake are of biosimilars around the country. And this is just an example study, an example figure, but we will look at, for example, census in Wales, in Scotland, anywhere we have data from BADBIR. And if we could, we will try and triangulate some of that data with other regional data to see how accurate our data is. Really, this is a descriptive study to help us understand the distribution of practice in the UK. The second question goes to the kind of the crux of our Dr. Strange question, which is trying to make sure that we have 2 similar cohorts so that we can ask the question, what if the patient started on this compared to this? And this is a schema just to demonstrate what we need to do. And really what we need to do is try and compare apples with apples instead of apples with oranges. And this is to make sure that those different cohorts, so people starting or switching on similar, are similar to those on reference products. And this is because there may be factors that are associated with the person rather than the drug, and we need to control for these in an observational study. So here we're going to make sure that patients have a stable use of reference products. So everybody switching to, let's say, Amjevita have to have had 6 months of a lead-in phase of the reference products for the switching cohort, and then we then compare them to the same or similar group of patients who've also had 6 months of the originator product. And here we're really trying to make sure that we answer the question and we don't get misled by factors that may be more associated with individual patients. We want to look at the population level to understand that what-if question. And finally, we're going to use a last study. So this is more of a complex study, but what we really want to do is to use all of the data we collect. And this kind of complex study design allows us to really utilize everybody, all the data in BADBIR. So if I just kind of talk you through some of this data, for let's say this patient A has started on Humira, and they started on Humira and they continue on Humira, the first line until the first blob. And this is the time where they switch onto Amjevita, let's say. What we want to do is to then compare that patient to all of these other patients who've also had 6 months of Humira. And then we could use this following time to then use to compare the follow-up from this blob onwards to all of these other follow-up times with the lines and the X's. And these patients may then go on to switch to a biosimilar as well, in which case they will drop out of the comparator, and we will use another group of patients to compare to the second blob, for example, and the third blob. So this means that we're using all the data, all the follow-up data, and not wasting any of it that we would, for example, waste a little bit in study 2. So we hope that these studies would really answer the question for patients. For example, a lot of patients tell us anecdotally when they switch, they seem to worsen. And we want to know, is this an individual effect? Is this a population level effect? Is this an effect associated with particular brands of biosimilars? Or is this something just, that actually would have happened anyway had the person continued the Humira? So with that, that is the study plan. I'd like to conclude again by thanking the Psoriasis Association. We're really grateful for the support. We couldn't start to do this without support, and we are now actively arranging the advert to recruit the PhD student to start working from October this year. If you have any questions or suggestions about the study, feel free to email me. I can take a photo of that email address, and you can reach out to me by DM on Twitter, or just tweet at me. If you are a patient on one of these biosimilars or even a reference product, it's really important, especially if you're on bad beer, that you remember to ask your doctor to write down the brand name on all the documentation in the clinic notes, in the letters, so that we really have a good idea of who is on what, so that we can do this type of study. So with that, I'd like to finish my talk. Thank you very much for your attention.